Hello learners, and welcome to the next part of our lecture about aqueous ionic equilibrium. So you know what that means. New part, new pre-lecture. So you got to go ahead and create your one-pager to orient your brain. You got to go ahead and review any previous chapter information and of course complete a practice problem to really get that concept down. And the goal is that the lectures are going to enhance or add to the pre-lecture information. So making that connection in this small step really does help in layering information and approaching the problem later on, folks. So let's get to thinking. So we have already seen the importance of aqueous solutions, like in Chapter 4 from Chem 101, where we learned about different types of chemical reactions. Or recently with Chapter 13 and 14, where we learned about the equilibrium and acids and bases within reactions, right? We now turn our attention to two additional topics involving aqueous solutions. Buffers, solutions that resist pH change, and in the next part of this chapter, solubility equilibria, which is how far a slightly soluble ionic compound dissolves in water. So let's first focus on buffers. And of course, we're going to be using some of our OG chemical Avengers to get us through this concept as we will revisit strong and weak acids and bases throughout this lecture portion, okay? Now back to buffers. Buffers are crucial in biology and chemistry. Why? Because they help maintain a stable pH level. Why is that going to be essential in biology? Just think of human beings. We have buffers already in our blood to maintain its pH, right? And maintaining a stable pH level is essential for preserving the proper functioning of biological systems. And of course, we use buffers in ensuring the reliability of chemical reactions, like for our chemical kinetics lab or our equilibrium labs, right? To give you an example, say we have a buffer solution here that has a pH of 5.00. If you add acid or 1 molar HCl, the pH goes down to 4.98. If you add NaOH, which you can't really see here, but it's going to be a strong base here, then your pH is going to increase to 5.02. And so that's a very slight change, which is why having a buffer is going to help us maintain a range of a pH. All right, folks? Now, going further, we're going to learn about acid-base titrations again, but we're going to go further and learn about acid-base titration curves. And that's going to represent the range of pH changes during a titration, and we're going to actually get into detail about different types of titration curves as well. And some of these same acid-base titration curves you will see for yourself in experiment 18 as we go further throughout the semester. And again, our OGs are going to come back for us, so let's put this all together and get into this part. To organize ourselves, we got to make sure that we have our little list here, right? And again, we're going to be using our OG chemical vendors to help us answer questions. What questions are we asking? We're going to learn about buffers. We're going to learn about how they maintain pH. We're going to learn about how to make calculations involving buffers easier with the Henderson-Hasselbalch equation. We're going to learn about buffer capacity and range and also how to prepare one in lab. And then we're going to end with acid-base titration curves. We're going to talk about strong acid and base titration curves, weak acid and strong base titration curves, weak base and strong acid, and even weak polyprotic acids. All right, folks? And to wrap up this part, we will be discussing acid-base indicators. So let's get this party started, folks.